Lotus Academy of Diet and Fitness.
Chapter 5 Lipid Metabolism. Lipid Metabolism. Lipid metabolism involves the synthesis, breakdown, and use of fats in the body. Fats are crucial for energy and cell structure. Here are the key aspects of lipid metabolism. Lipid digestion. During digestion, dietary fats are broken down into simpler molecules. Bile salts in the small intestine emulsify fats, making them easier to process. Pancreatic lipases then break triglycerides into free fatty acids and monoglycerides. Absorption of fatty acids. Free fatty acids and monoglycerides are absorbed by the intestinal cells, enterocytes, lining the small intestine. Within the enterocytes, these molecules are reassembled into triglycerides. Formation of lipoproteins. Triglycerides, cholesterol, and fat-soluble vitamins combine with proteins in the enterocytes to form chylomicrons. These are large lipoprotein particles that transport dietary fats from the intestine to tissues via the lymphatic system and bloodstream. Lipoprotein metabolism. Chylomicrons, along with other lipoproteins like VLDL, LDL, and HDL, play roles in transporting lipids throughout the body. VLDL transports endogenously synthesized triglycerides from the liver to other tissues. LDL carries cholesterol from the liver to peripheral tissues, including the cells that require cholesterol for membrane synthesis. HDL helps transport cholesterol from peripheral tissues back to the liver for elimination or recycling. Lipolysis. Lipolysis breaks down stored triglycerides into free fatty acids and glycerol, mainly in adipose tissue. Hormone-sensitive lipase regulates this process, releasing fatty acids for energy use in different tissues. Fatty acid oxidation, beta oxidation. Fatty acids are broken down via beta oxidation in mitochondria. Enzymes remove 2 carbon units from the chain, yielding acetyl-CoA. This enters the citric acid cycle to produce ATP. Lipogenesis. Lipogenesis creates new lipids like triglycerides from extra carbs or energy. It happens in the liver and fat tissue using acetyl-CoA from carb or amino acid breakdown. Ketogenesis. Ketogenesis occurs in the liver when carbohydrate supply is limited. Ketone bodies like acetone, acetoacetate, and beta-hydroxybutyrate are produced. They serve as alternative energy sources, especially for the brain and muscles during fasting or low-carb diets. Lipid storage. Excess dietary fats and newly synthesized triglycerides are stored in adipose tissue as lipid droplets. Adipose tissue serves as the primary site for long-term energy storage. Formation of lipoproteins in lipid metabolism. Lipoproteins are essential for lipid transport in the body, carrying cholesterol and triglycerides. They consist of a lipid core surrounded by proteins, phospholipids, and cholesterol. These particles package and shuttle lipids among different body tissues and organs. Here's an overview of the formation of lipoproteins in lipid metabolism. Synthesis of polypoproteins. Polypoproteins are crucial structural components of lipoproteins. They also act as ligands for lipoprotein receptors and enzymes in lipid metabolism. Synthesized in the liver and intestinal cells, polypoproteins include ApoA, ApoB, ApoC, and ApoE. Assembly of lipoprotein particles. In the liver and intestine, lipids combine with apolipoproteins to form lipoprotein particles. Different combinations of apolipoproteins give rise to various types of lipoproteins, such as VLDL and HDL. VLDL assembly and secretion. VLDL, synthesized in the liver, transports triglycerides and cholesterol esters with ApoB 100. It delivers endogenous triglycerides to tissues. Conversion of VLDL to LDL. VLDL circulates, undergoes lipolysis by lipoprotein lipase, LPL, forming IDL. Continued triglyceride removal transforms IDL into LDL, the main cholesterol carrier. HDL formation. HDL or good cholesterol forms in the liver and intestine by combining APOAI with free cholesterol and phospholipids. It aids in reverse cholesterol transport, moving cholesterol from tissues to the liver for disposal. Lipoprotein metabolism and remodeling. Enzymes like LPL, hepatic lipase, and LCAT remodel lipoproteins as they circulate in the bloodstream. This affects lipid composition, size, and lipid transport to tissues, regulating cholesterol balance and impacting cardiovascular health. It's important to note that imbalances in lipoprotein metabolism, such as elevated LDL cholesterol levels or low HDL cholesterol levels, are associated with an increased risk of cardiovascular diseases. Lipolysis in lipid metabolism. Lipolysis breaks down stored triglycerides in adipose tissue into glycerol and free fatty acids, which serve as energy sources for tissues. This process primarily occurs in adipocytes, releasing fatty acids for energy utilization. Here's an overview of lipolysis in lipid metabolism. Activation of lipolysis. Hormones like epinephrine, norepinephrine, glucagon, and growth hormone regulate lipolysis. They bind to adipocyte receptors, triggering a cascade that activates the breakdown of triglycerides into fatty acids and glycerol. Activation of hormone-sensitive lipase (HSL). Hormone-sensitive lipase (HSL) is activated by hormonal signals through phosphorylation, converting it into an active form. Activated HSL can then access stored triglycerides in adipocyte lipid droplets to initiate lipolysis. Hydrolysis of triglycerides. Hormone-sensitive lipase catalyzes the hydrolysis of triglycerides into glycerol and free fatty acids by breaking ester bonds between the glycerol molecule and the fatty acids. This process releases fatty acids from the triglyceride molecule for release into the bloodstream. Release of free fatty acids. Once liberated from adipocytes, free fatty acids enter the bloodstream, binding to albumin for transport to different tissues. Their concentration is tightly regulated in the bloodstream, influenced by hormone levels, nutritional status, and metabolic demands. Uptake and utilization of free fatty acids. During lipolysis, free fatty acids are released and taken up by tissues like skeletal muscle, liver, and cardiac muscle, serving as an energy source. They enter cells through fatty acid transporters and undergo oxidation in the mitochondria to produce ATP. Glycerol utilization. Glycerol, another product of triglyceride breakdown, is released into the bloodstream during lipolysis. It's taken up by the liver and other tissues, where it can be converted into glucose via gluconeogenesis or used for energy production. Regulation of lipolysis. Lipolysis is tightly regulated by various factors. Alongside hormonal control, insulin inhibits lipolysis, while intracellular levels of cyclic adenosine monophosphate (CAMP) activate hormone-sensitive lipase, influencing the process. Lipolysis is vital for releasing stored energy in the form of triglycerides, enabling the body to use fatty acids as fuel during energy-demanding periods like fasting or exercise. Yet, uncontrolled lipolysis can disrupt lipid metabolism, potentially leading to metabolic disorders. Lipid storage in lipid metabolism. Storing fat is important for our body's energy needs. It puts away extra fats as triglycerides in fat cells for later use. Fat cells, found mainly in fat tissue, are where this storing happens. Here is an overview of lipid storage in lipid metabolism. Triglyceride synthesis. Excess dietary fats or lipids, along with those synthesized through processes like liver lipogenesis, convert into triglycerides for storage. In lipogenesis, fatty acids derived from non-lipid precursors such as glucose combine with glycerol 3-phosphate to form triglycerides. Packaging into lipid droplets. Triglycerides, once synthesized, are encapsulated within lipid droplets in adipocytes, the main cells of adipose tissue. These droplets feature a core of stored triglycerides ensconced by a monolayer of phospholipids, cholesterol, and related proteins. Regulation of lipid storage. Lipid storage is regulated by hormonal signals, nutrient availability, and energy balance. Insulin promotes lipid storage by enhancing fatty acid uptake into adipocytes and stimulating triglyceride synthesis. Conversely, hormones like glucagon and adrenaline trigger lipolysis, releasing stored energy by breaking down triglycerides into fatty acids. Adipocyte differentiation and expansion. Adipose tissue growth and lipid storage depend on adipocyte differentiation and expansion. Differentiation converts pre-adipocytes into mature adipocytes regulated by transcription factors and signaling pathways. Expansion occurs via hypertrophy, enlarging existing adipocytes, or hyperplasia, generating new adipocytes from pre-adipocytes. Lipid mobilization. During periods of energy demand like fasting or increased activity, stored triglycerides are mobilized from adipose tissue via lipolysis. Hormonal signals like glucagon and adrenaline trigger lipolysis, breaking down triglycerides into glycerol and free fatty acids. These fatty acids serve as an energy source for various tissues. Lipid remodeling and turnover. Lipid storage is dynamic, involving continuous remodeling of triglycerides within adipocytes. This process regulates lipid droplet size and composition. Triglycerides can be broken down through lipolysis for energy or resynthesized through lipogenesis for storage. Adipose tissue plays crucial roles, serving as an energy reservoir, insulating organs, and producing hormones for metabolic regulation. It also generates signaling molecules impacting inflammation and satiety, highlighting its importance in overall health. Disrupted lipid storage and adipose tissue function can result in metabolic disorders like obesity, insulin resistance, and dyslipidemia. Maintaining proper regulation of lipid storage and turnover is essential for metabolic balance and overall health. The impact of lipids on cardiovascular health and inflammation. Lipids are central to cardiovascular health and inflammation through lipid metabolism, governing lipid synthesis, transport, utilization, and storage. Recognizing their influence is pivotal in preventing and treating cardiovascular diseases and associated inflammatory disorders. Cardiovascular health. Cholesterol, vital for cell functions and hormone production, can pose risk when LDL cholesterol levels rise, causing artery plaque buildup and raising atherosclerosis risk. This condition heightens susceptibility to cardiovascular ailments like coronary artery disease, heart attack, and stroke. Triglycerides, storing excess dietary energy in fat cells, elevate cardiovascular disease risk when blood levels are high, especially alongside low HDL cholesterol, termed good cholesterol. HDL cholesterol, termed good cholesterol, aids in removing excess cholesterol from arteries, returning it to the liver for disposal. Elevated HDL levels are linked to decreased cardiovascular disease risk. Inflammation and lipid metabolism. Inflammatory response. Inflammation, a natural immune reaction to injury or infection, can exacerbate cardiovascular diseases if chronic lipids, notably oxidized LDL cholesterol, may prompt arterial wall inflammation, resulting in foam cell formation and atherosclerotic plaque genesis. Adipose tissue. Excessive fat accumulation in obesity prompts adipose tissue to produce more pro-inflammatory cytokines and adipokines. This chronic inflammation can foster insulin resistance and metabolic syndrome, raising the risk of cardiovascular diseases. Role of diet and lifestyle. Diet. High saturated and trans fat diets raise LDL cholesterol, promoting inflammation and cardiovascular risk. Diets rich in unsaturated fats like omega-3 from fish, nuts, and seeds reduce inflammation and improve lipid profiles. Exercise. Regular exercise boosts HDL cholesterol, lowers triglycerides, and improves heart health.